Test, test, test. Test, test.
Glimpses.
Hello, do you hear me? Do you hear me, Elvis?
Yes, it's clear.
Oh, good, good. Thank you, thank you.
Hi, Moriaki. Hi.
Hi, how are you?
I'm good. I just send my kids to dentist. Then I come back to host your seminar.
Oh, I see, I
Yes.
see.
Yes.
Good.
Thanks
good.
a lot for giving us our journal audience this seminar. And let's wait to see 5 more minutes, 4 or 5 more minutes, then we can start. I guess this is on different platform, including some Chinese platform, and it's live. So maybe a lot of other audience is also watching us.
Nice.
Yeah, but because I'm at home, so I'm not showing the video. Just you know, it's not that stable. My internet at
Sure.
home. Yeah. Yeah, but we can see you very clear. That's great.
Good, good.
And where are you, Moriaki? I saw you are attending some conferences somewhere.
Yeah, I was in Thailand last week
Okay, okay,
for a paleontology conference, but just came back to Copenhagen.
okay, okay. Yeah, I saw you were posting some Twitters. Yeah,
Mhm. Yeah,
yes,
I'm here, like you
yeah,
see,
beautiful.
Copenhagen.
Yeah, and it's getting colder a little bit here now, right?
Yeah, now, yeah, finally, a bit
Yeah,
cold,
a bit
especially
cold. Yeah.
just coming back from Thailand. It's still
Oh,
like
exactly,
summer,
exactly.
so
Yeah. Let me see who uh, is there any editor who is also hosting this, or it's just me? Let me check.
sure.
I see Kenny. Hi, Kenny. Are you are you there, Kenny? We haven't started yet, so just want to say hi to you if you can hear us. Yeah, so the reason I think the reason why there's not many attendance here in Zoom is because a lot of audience, I mean Chinese audience, they are using different platforms, so we can't see who are there. But they will post questions on different platform, and they will be directed to me. So in case of any questions we don't see here, and I guess they will send to me, and I will just deliver to you later.
Sure. And I guess
Yeah.
we panelists can speak, and probably normal attendants like Kenny cannot speak here, I guess.
Oh, yeah, maybe.
Yeah, yeah.
Yeah, maybe, maybe. I think, yeah, they are muted. Um, okay, so I think now is time. Almost
Mhm.
2 p.m. in Denmark and 9 p.m. in Beijing. So welcome everyone, and this is the talk at Eco-Environment&Health, so the journal talk series. And today we are so glad to have Professor Dr. Moriaki from the University of Hong Kong. And on behalf of the editorial board, I really welcome Moriaki and give us this very inspiring, you can see the topic, time machine biology. Paleobiology provide glimpse of future oceans. So let me give you a brief introduction of Moriaki. And we know each other for over 10 years. And that time I was a PhD student, and Moriaki started his professorship in Hong Kong. And Moriaki is a paleobiologist and associate professor now at the University of Hong Kong. And his recent research has focused on the tropical biodiversity, the spatial temporal dynamics of large-scale biodiversity patterns, and their machines, mechanisms behind. And he is also interested in microfossils based conservation, paleobiology, and paleontology of the Austracoda in general. Recently, he has been also developing a new research directions, deep learning based automation, really a new topic in micro paleontology. So please, Moriaki, the time is yours.
Yeah, thank you, Elvis. Thank you a lot for the great introduction. So let me share the screen. Yeah. Then, yeah. So today I'm talking about time machine biology. So I recently used this term for paleobiological research to understand past biodiversity, past ecosystems, and climatic impact, especially using sediment cores and small fossils. So I will introduce this a bit as a start. And I guess many of you are not a geologist or paleontologist, so this cartoon will be great to briefly explain what we do usually. So we use sediment core. It's a kind of cylindrical section, like tube-like section of sediment. And sediment is gradually accumulating. So that means surface sediment is present and deeper sediment is deeper past. So by getting sediment core like this, a cross section of sedimentological history, we can understand long-term time series changes in like biodiversity or environments. So we go to the ocean by research vessel and get sediment core and cut into half and slicing into every centimeter. And then by studying each subsample one by one from the bottom to the top, we can reconstruct the history of our ecosystem or biodiversity. So we have simple procedure, just wet sieving to remove muddy materials and to concentrate microfossil specimens on the sieve, and using stereo microscope to pick and count and identify species and specimen. This is basically what we do. The sediment core is great because they have really good age model, so we can know the precise age of sediment in sediment core, so we can know or reconstruct detailed high time resolution history over hundreds or thousands or even millions of years. But one weak point of sediment core is it's small, like a diameter of sediment core is usually 10 centimeter or smaller. So we cannot study like a big fossils like a dinosaur or ammonite. It's too big to study based on small sediment available from sediment core. So we need to study something small, but luckily we have a bunch of small fossils, so-called microfossils. That means microscopic size fossils. We need microscope to observe them. So they are both plankton and benthos, and also different taxonomic group. For example, I'm ostracod specialist, is tiny crustacean, benthic animals, and we have other protozoan or unicellular organisms like benthic and planktic foraminiferans, and the coccolosphere, diatom, radiolarian, and so on. So they are quite highly variable type of small organisms are available in sediment core. We can get like 100 or 1,000 or even more specimen of type of microfossils from just a small amount of sediment sample. So I'm an ostracod specialist myself, so I most often use ostracod as a proxy to understand deep time biodiversity. And it's abundant benthic microfossils, and they have high biodiversity. They have many species, and they're probably the only multicellular animals or metazoan organisms. They are abundant enough in sediment core, and because they are crustaceans, tiny crustaceans with good shells, well preserved in fossils. You can imagine biodiversity. For example, this is a picture from Antarctica, Southern Ocean deep sea, showing beautiful biodiversity in deep sea organisms. And we have like octopus, sea spiders, brittle stars, urchin, and so on. But if we study deep time sediment core paleoecology, we don't have most of them. Most of them simply don't have hard parts preserved as fossils, and some are a bit big, so they have low chance to have high abundance in sediment core. So only microfossils like foraminiferans or ostracod have a high abundance in sediment core, and so usable for time machine biology. So we can get hundreds of specimens, and we can use some reasonably robust statistics to understand biodiversity and their history. So we need to study something small, so-called microfossils. So microfossils in sediment core is quite important, not only to self, but also as a kind of window to look into the deep past ecosystems and biodiversity. Because as I mentioned in the last slides, most of organisms are not well preserved as fossils. So we have no much choice. We need to use microfossils as a proxy or representative of broader marine community or biodiversity. Then we can look into the past. For example, how many species are there, how healthy was ecosystems at that time, and so on. And as Elvis introduced me really nicely, I'm in Hong Kong. And I moved to Hong Kong 2011, January. And after that, I've been in Hong Kong more than 10 years already. And I'm quite lucky to work with fantastic students and also postdoc people to work together on time machine biology or micropaleontology or Australcott micropaleontology. And one of our main research topic is climatic impact on marine ecosystems and biodiversity. So today I'm talking about two research topics related. One is temperature as a main driver of marine biodiversity. I've been working on this topic quite a long time. I started this already when I was postdoc. So still I'm interested in and working on more than 10 years. And the second one is I'm based in Hong Kong. So I'd like to introduce Hong Kong paleobiology. Hong Kong is quite interesting place, high biodiversity in the tropics, affected by both climatic change and pollution or human-related impact. So I'd like to introduce subtidal ecosystems there as the second topic. So let me start with temperature. Temperature drives marine biodiversity. I tend to think yes, so I try to explain. And in biology field, at least global scale in the ocean, we have increasingly better understanding or consensus temperature as the main driver of biodiversity, at least at global scale and in the ocean. And this is a result of Census of Marine Life project about 10 years ago. It's a project to compile and to include all available species data, occurrence data to understand biodiversity of our ocean as much as possible, including all available species and taxonomic group into one database. And the result is this heat map of biodiversity. So orange, red colors mean higher diversity, that mean more species, and bluish color mean less biodiversity or less species. So in this map, we clearly see latitudinal diversity gradient. It's a probably most fundamental or most common pattern, ecological pattern on our planet. And biodiversity is higher in the tropics, like a coral reef, and much lower in higher latitudes, like Arctic Ocean or Antarctica. So we see reddish color, like orange, red in lower latitude in tropics, and we have more bluish color in the Arctic and Antarctica. So we see latitudinal diversity gradient pretty clearly. And they conducted simple regression modeling, model selection, and they consistently indicate temperature as the best predictor of global ocean biodiversity patterns. And if we plot diversity as vertical axis and temperature as horizontal axis, we have indeed nice correlation between diversity and temperature. But if we look into a bit more details, it can be a bit more complicated. This is our recent study with great collaborators listed here about the plankton biodiversity, zooplankton biodiversity. In this case, it's not ostracod. We use planktic foraminiferans. And this is a relationship between species diversity as y-axis and SST, sea surface temperature, as horizontal axis. So we see latitudinal diversity gradient or positive temperature diversity relationship. So temperature is getting warmer, we have more species. It's the same as lower latitude have more species and higher latitude have less species with temperature decline along latitude. But if we go certain level, we have threshold. If temperature go up beyond like 25 degrees Celsius, the relationship with diversity turned into negative. So too hot temperature affect biodiversity negatively. So if we see the temperature diversity relationship closely, it's a sort of unimodal. We have peak at the 20 to 25 degrees Celsius, and higher than that temperature have a negative impact on diversity. So that means too much temperature affect biodiversity negatively. So too hot is not so good for biodiversity. And this is the same data showing in different way. So this is biodiversity heat map like before. So reddish color mean more species and bluish color mean less species. And this is latitudinal diversity gradient. So vertical axis is latitude. So 0 mean equator and positive number mean northern hemisphere and negative numbers mean southern hemisphere. And horizontal axis is species richness. So these curves mean latitudinal diversity gradient. So by looking at heat maps, we still see latitudinal diversity gradient. Like we see more reddish, orange, red colors in low latitude in tropics, and we have more blue colors in higher latitude in Arctic and Antarctica. But if we see latitudinal diversity gradient curves, it's a bit different in details. We have 3 time slices by using paleontological data, climatic modeling, and future projection. So the top panel is last glacial maximum or last ice age. It's about 20,000 years ago. And the middle panel is kind of present day, modern but pre-industrial time. And the bottom one is future projection based on IPCC scenario, business as usual. That means it's based on the scenario when we don't care about the CO2 emission at all in the future. So in the last ice age, we see normal latitudinal diversity gradient and biodiversity is highest in the lowest latitude at the equator. But in the present day, temperature is already too hot in the equator, and we have clear, small but still clear diversity dip or decline in the equator. And this diversity dip will be much stronger in the future if we didn't reduce any CO2 emission. So present day temperature is already too hot for tropical biodiversity, and it will be more so in the future if it were much warmer than now. So this data clearly indicate tropical ocean is already too hot for marine biodiversity and will be more so in the future. And this is the comparison of 2 time slices regarding biodiversity. So biodiversity change from last ice age to present is this panel. And the mid panel is the time change from present day to the future. In business as usual scenario, we didn't reduce any carbon dioxide emission. And this panel is also present to future, but in good scenario when we reduce CO2 emission very much in the future. And so plus numbers mean species increased from one time to the other time, and minus means species richness decline from one to the other time slice. So from last ice age to the present, we see tropical diversity decline here. And such species will go up to higher latitude to track their temperature niche or thermal niche. So tropical temperature going to be too hot after last ice age warming, climatic warming into the Holocene. Tropical species escaped into mid-latitude. It's more comfortable for them. And present to the future, we have much stronger diversity decline here in the equator, and they also escaped to mid to high latitude. And it's a bit interesting or even scary because this one is last ice age to present. That means about 20,000 years. But this one is present to future, just 100 years. But still, the tropical diversity decline is much stronger than this. So our ongoing diversity decline in the tropical region is quite substantial and will be so in the future. But the one good news is if we reduce carbon dioxide emission nicely, we can minimize such change in the future. So this is a cartoon we recently published based on not only this particular research, but also combining all available paleobiology data and contemporary observation, fishery data, biological data, and so on to have my idea or big picture of past, present, and future ocean biodiversity. So this is the past, and this is present, and this is future. And we have different latitudes. So bottom is tropical latitude and temperate latitude in the middle, so middle latitude. And the top panels mean polar or Arctic latitudes. And from past, present, future, we have global warming, temperature is going up. And we have three types of animals. It looks like fish, but it's not necessarily fish. It's just cartoon or general representation of organisms. So red ones are hot water species or hot water adapted species. And orange ones are intermediate or warm water adapted species. And the green one or blue one is cold water adapted species. So from past to present to future, temperature is going up and tropical ocean is going to be too hot, even for hot water adapted species. So they escape from the tropics and moved to higher latitude gradually to present and to the future. Similarly, warm water fish also track their thermal or temperature niche going further up to the north or higher latitude. And cold water species shown by blue, they are unlucky because it's already highest latitude. So they cannot escape to colder place. It doesn't exist. So they may decline in the future, and they may have much higher risk of extinction than other species. And one important thing is future tropics. It's going to be empty if we don't reduce any CO2 emission, because it's too hot, even for hottest water adapted species. And it's already hottest adapted species existing on our planet. So there is no replacement into tropical region in the future. So if we continue this normal business, we have substantially different tropical ocean ecosystems with much less species. So this cartoon and general conceptualization clearly show it's important to combine both paleobiological data, time machine biology data with contemporary biological and ecological data. Because paleobiology is not only important to understand the past itself, but also especially by incorporate with other data in biology and oceanography, it's quite useful to understand present day situation and also better prepared for coming future. So this is the first part of my talk. And let's moving on the topic about Hong Kong. So I'm on sabbatical right now in Copenhagen, but I'm based in Hong Kong, University of Hong Kong. And Hong Kong is not always like this, but at least downtown Hong Kong is super urbanized with skyscrapers, super high population density and local pollution and light pollution, and so on. And it's history. And this is the cartoon over the historical picture. So this background black-white picture is British colonial time, maybe 100 years ago. It's already big city, urban, relatively urban, but it's not as like a present-day skyscraper Hong Kong. So after that, we have rapid development, having more buildings and land reclamation, etc. So they rapidly changed Hong Kong into the modern gigantic city or metropolis. So I'm interested in history of Hong Kong, especially regarding marine ecosystems. So this project is led by my former student and current postdoc, Yuan Hong, and with great collaborators, Paul Harnick, and my former postdoc, Hokuto Iwatani, and statistical analytical people, An Chao and Chi-Ling Wei. They did a great job with statistics and analysis. And we have really good collaboration, and we still continue our collaboration, and we enjoy pretty much. So after my moving to Hong Kong, I've been doing Hong Kong time machine biology or Hong Kong conservation paleobiology. Especially recently, we have a good bunch of funding to get new sediment and new sediment core. So we try to understand young history of Hong Kong ecosystem, especially regarding climatic impact and pollution impact, because Hong Kong is quite interesting situation, both affected by urbanization, pollution, and also climatic change, such as Asian monsoon. I will explain later. So we get sediment core, surface sediment, and process as I shown in the previous cartoon, and we identify ostracods mainly, and we use ostracod as a broader representative or paleoproxy to understand broad past Hong Kong ecosystems and biodiversity. So in this time, we got surface sediment samples from a bunch of localities in Hong Kong. And this is the same locality as Hong Kong Environmental Protection Department's environmental monitoring site. So it's a really good advantage. Hong Kong have really good environmental monitoring program in all these sites, and we have all environmental data available online. So we can directly compare biological or ostracod result with environmental parameters. And in this case, we liked to understand spatial distribution. So we didn't use sediment core, but instead we use two time slices in grab sample. One is the surface sediment. It's probably past 5 years or more or less present day. And then the subsurface sample in the below the surface, and it's the past. In considering Hong Kong subtidal sedimentation rate, it's probably past 50 to 100 years ago. So we are comparing two time slices. One is the present day, and the other is the past, about 100 years ago. We use hill numbers for biodiversity measures, and it's increasingly more popular both in ecology and paleoecology, and they recommend to use hill numbers recently. It's quite reasonable. And we have three types of hill numbers, so-called Q equals 0, 1, 2. So Q equals 0 means species richness, so more or less presence-absence based diversity. And Q equals 1 is kind of Shannon. They reflect more abundant species proportion. And Q equals 2 is more like Simpson index. They consider very abundant species more. So this is biodiversity in each region divided by environmental monitoring program. So it's not biological division, but the Hong Kong government division. And the dotted line means subsurface, so it's the past. And the solid line is the present day, so surface sediment. And the vertical axis is species diversity or richness, so higher number mean more species or higher diversity. And this is the order Q, like Q equals 0, 1, 2, so different diversity metrics in Hill numbers. We don't see much clear trend in regional scale. For example, in some region, we have much higher diversity in the past, and the present day much lower biodiversity. But in several sites, it's quite similar between past and present. And in some case, present day diversity is higher than the past. So we don't see much consistent pattern, at least by looking regional level biodiversity. So obviously, we need to look into more details. So we calculated beta diversity or faunal turnover from the past to present. So this is a faunal difference between subsurface sample and surface sample. So it's a faunal change from the past to the present in 3 different metrics, Sorensen and Horn and Morishita Horn. It's same as Hill number Q equals 0, 1, 2. So Sorensen means presence-absence based turnover. And the Horn is a turnover based on more abundant species. And Morishita Horn is based on very abundant or dominant species. So we see much clearer trend than regional diversity comparison in the last slide. In the presence-absence data, so that means it reflects more rare species. We see clear increase in species turnover from the west to the east. So eastern water have much bigger faunal turnover in rare species. And abundant species and very abundant species turnover is a bit unclear, but we see higher turnover in central Hong Kong and much lower turnover in either western waters and eastern water. This is the same data, is showing in different way. You may see this right-hand side panel may be clearer than the last panel. So in rare species turnover, we see clear increase from west to east. And we have central Hong Kong, higher turnover in abundant species and dominant species metrics. So we have different trend between rare species and abundant species. And why? Or what is the in charge factor for this trend? So we conducted simple regression modeling and model selections with available environmental parameters in Hong Kong Environmental Protection Government's monitoring data. And for rare species turnover, we see significant result with like turbidity, mud content, salinity. So we tend to think river discharge, Pearl River discharge is in charge for this west to east increase in faunal turnover. I will explain more in the next slide. And regarding abundant species and very abundant species, the same modeling result indicate metal pollution as the dominant predictor of the patterns. So central Hong Kong rapid or strong turnover in abundant species and very abundant species is probably because or related to pollution, metal pollution. And Pearl River influence on rare species turnover, it makes sense because Hong Kong is pretty much influenced by Pearl River discharge. And Pearl River is one of the biggest river in Asia or in the world, and it's situated in the western part of Hong Kong, somewhere here outside of the map. And they have strong freshwater discharge this side from west to east. So western side is more brackish water or estuarine-like environment in Hong Kong. And eastern side is characterized by higher salinity and it's more oceanic. And it is known that Asian monsoon have been changing historically, and about 100 years ago, Asian monsoon was much stronger. So that means more precipitation, and the Pearl River discharge probably reached to much more eastern part than present day. And I tend to think eastern water, submarine, subtidal ecosystems are more sensitive to such a little bit changes in salinity, turbidity, and the mud sediment availability, because western water is always estuary and always dynamic, changing in salinity in different seasons, and often it's quite low and turbidity is quite high. So they are already adapted to such a bit difficult situation of changing salinity, etc. So they are more resistant. But oceanic side in the east is more adapted to normal marine. So they are not so well prepared or resistant or changes in freshwater related environments like salinity decline and so on. So when Asian monsoon was much stronger, probably eastern side ecosystems have much more impact than western water. This is my interpretation of this result for rare species. And for abundant species and dominant species, metal is responsible. So there are two possibilities. One is local pollution, because central Hong Kong is like this, super urbanized and high population density. So we have a bunch of local pollution. And the other possibility is anti-fouling paints, because Hong Kong is busiest port, some of the busiest port in the world. And also metal is always included in anti-fouling paint like copper. So either of local metal pollution or ship traffic related metal pollution from anti-fouling paint, or both, could be responsible to strong turnover of abundant and dominant species in central Hong Kong. So this result is not only interesting, particularly for Hong Kong, but also in general to think about tropical future with human being. Hong Kong is situated in here, northern tip of Coral Triangle, biodiversity hotspot. So biodiversity is pretty high in Hong Kong. And as I mentioned earlier, Hong Kong is pretty urbanized, so suffered by pollution, and also next to Pearl River, we have a bunch of climatic impact. So this combination is interesting, high biodiversity in the tropics and suffered by both pollution and future climatic change. It's a kind of future of many other tropical biodiverse regions, not yet suffered by too much human impact or too much climatic change related to carbon dioxide emission, etc. So Hong Kong is really interesting case study. We are already experiencing the future of many different places. So it's a reason why I'm interested in Hong Kong conservation paleoecology, and to understand recent fossil record, to understand Anthropocene changes. And I have a few more slides about my ongoing or future research interest I like to share. As Elvis introduced, I'm pretty interested in tropical biodiversity in long time scale, entire Cenozoic, like past 66 million years. And as I explained, latitudinal diversity gradient, similar heat map here showing higher tropical biodiversity. But even within tropics, biodiversity distribution is not homogeneous. It's quite different. And Coral Triangle here, Indonesia, Philippines, etc., have a super high biodiversity compared to any other equatorial region like Africa or Caribbean Sea. They have much lower biodiversity compared to Coral Triangle. And I'm interested in such a coral triangle have been persistent for the past about 20 million years. Their biodiversity have been continuously high for a long period. But beyond that, biodiversity hotspots have been changing in the place, some paleobiological research indicate. So I like to know more detail about such, especially related to the changes in temperature and also changes in continental arrangement. This is a sea surface temperature heat map from the past 40 million years ago to the present. And also we have different continental arrangement by plate tectonics. So this is present day. And 40 million years ago, Eocene was much warmer, tropics, as shown by much reddish, darker reddish color here compared to the present. And also continental arrangement was quite different. Like Australia was close to the Antarctica, and India was still island, not yet attached to the Eurasian continent. And we have big Tethys Ocean, it's a present-day Mediterranean, but at that time, it's a huge tropical ocean connected not only to Atlantic Ocean to the west, but also to the Indian Pacific Ocean to the east. It was the biodiversity hotspot at that time. And also, as Elvis mentioned, I'm recently interested in deep learning-based automatic identifications. And we are lucky to buy Keyence digital microscope in the University of Hong Kong. And it have a nice fully mechanical stages, X, Y, Z. So that means we can scan entire slide with all in focus with hundreds of specimens in one time. So we try to identify each individuals and identify each species and having census faunal count data automatic to have much bigger data than before and much more quickly. I tend to believe it leads to much bigger data before impossible and may be able to change the field. And I'm starting from ostracod because I'm ostracod specialist, but I'm interested in expanding to any other small things like not only microfossils, but also, for example, microplastics, as I a bit discussed with Elvis when I visited him in Denmark. And also now we have COP27 in Egypt. And I couldn't go, unfortunately, because I had a conference in Thailand, etc. But I'm involved with DOSI, Deep Ocean Stewardship Initiative. And I'm co-lead with Lisa Evin in Scripps about the climatic change group in DOSI. And we are particularly interested in green engineering technique impact into deep sea in regard with ecosystem functioning and ecosystem service and so on, shown here. For example, we have some engineering technology, like we can put CO2 in deep ocean as a liquid or solid phase, or we can get green electrical power by using temperature difference from deep sea to shallow marine. And it's important to fight against temperature increase and global warming ongoing and also in the future. But at the same time, we don't know almost anything about deep sea ecosystem impact from such engineering technique. So we need to be super careful at the same time. It's a bit complicated issue, but I'm quite interested in discussing such conservation issue. And me as a paleobiologist or time machine biologist, I'm keen to give long-term data and long-term perspective into conservation context. So if you are interested in any of tropical biodiversity and deep learning automation and deep sea conservation issue, I'm very happy to chat and discuss. And I think this is my last slide. And thank you for your attention. I'm very happy to take any questions.
Thank you so much, Moriaki. As always, excellent talk and covers a lot of hot topics, and many sit on the phone for fronts of multiple research areas: biodiversity, deep ocean, climate change. So now we have around 10 to 15 minutes. Of course, we can extend if there are more questions. So I think if you are in the audience, you can unmute yourself, just ask questions freely. Otherwise, you can also type in the chat, so we can see it and answer your questions. 白天老师，您在吗？是不是还有别的平台的一些问题，您可以帮忙传到我们这边，因为我们只能看到 Zoom 上的现在。
徐老师好，我在的。
OK， 谢谢。
可能还有一些延迟，现在其他平台上暂时还没有提问。
OK， 没问题。And by the way, please also allow me to introduce Professor Kenny Leung. I saw he's with us through the talk. Kenny, are you still there, and can you speak to us, or do you want to ask any questions or give some comments? I think you are the best to talk about Hong Kong. Don't know if you are able to unmute yourself though. Yeah, and then maybe I will ask the first question. Well, of course, Moriaki, we talked a couple of weeks ago already, but I have a new question related to your today's talk, because you add a lot of new stuff here. So I'm just wondering, you showed a lot of data and on the ocean. I'm just wondering whether this climate change thing will be bigger to the terrestrial system. You know, the lands get heat up much faster than the water, than the ocean, and get cooling down much faster than the ocean, than the water. So do you think there will be bigger impact on the terrestrial system using your similar ideas?
Yeah, great question. And it can be more complicated. I think both in the ocean and also terrestrial environment, there are a bunch of biological data already showing they are moving in responding to climatic change. But we may see more straightforward pattern in the ocean compared to the terrestrial, because ocean is filled by water. So we don't need to care about water availability, and also they can move or disperse relatively easily. But in the terrestrial environment, the atmosphere, so that means they need precipitation to survive. So they have not only temperature, but also precipitation and other related environmental factor as a critical environmental factor also related to future climatic change. And also some have really local distribution and they have difficulty to disperse or move. So tropical future could be a bit more complicated, I think.
I see. So now I just enabled everyone that can unmute yourself. If you want to ask directly, you just try to unmute yourself and speak up. I think we can hear you now. Otherwise, just type in your questions in the webinar chat. Yeah, maybe if I will just take this opportunity to ask a second question, then so in some of your maps, and also I attended some climate change seminars in the past few days. It seems a lot of efforts or data are showing the or predict the change on the Arctic area, but in the Antarctic area, it seems there are less studies or less data or less predicted impact. Do you have any ideas, or is it because of the data availability, or is it something else?
Yeah, data availability is quite critical issue. As you mentioned, polar area is more remote, so difficult to access. So we have less data, and it same is true for tropical ocean, because many, for example, developed country, United States, Europe are in higher latitude, but not in the tropics. So we need to take care of such possible data bias.
Okay, I see. I think it makes a lot of sense. Now we are getting questions flowing in now. Let me just read the first question. So how to judge the eastern water ecosystem in Hong Kong is vulnerable to climate change while western is not?
Yeah, it's based on our result, but also, for example, in the western water, they tend to have more like resilient species or resistant species to different types of environmental change. So we have a higher abundance of species with wide salinity range and also eutrophication tolerance, etc. And also Western water environment is quite dynamic in different seasons or different years, because Pearl River discharge is stronger in some season and weaker in dry season, and it changes with years. So I tend to think Western water system or Western water species or ecosystem is more resistant, and Eastern water species is more just adapted to normal-ish ocean environment. So if we have suddenly lower salinity or stronger river discharge impact, they could have bigger impact than Western water fauna.
Great. Then the next question is, can ostracod represent the whole biology, all creatures in the past?
I tend to say we see similar trend. Like ostracod is crustacean, a part of arthropods, and they show normal latitudinal diversity gradient similar to other or broader animals or organismal groups. So they are not contrarians. They are not the group showing unusual trend compared to other marine fauna. And so I tend to think the picture we can see in ostracod is generally similar in broader marine ecosystem, and it can be representative, I tend to think. For example, we have diversity increase with increasing temperature in ostracods just as many other marine organisms. So yeah, I tend to think so.
All right. Next question is from our friend, Professor Kenny Leung. And the question is, just have one question. 1 million, in millions years ago, we had a hotter ocean and there would be reduction of biodiversity, even site-specific extinctions, but the diversity did return and recover, as shown by the mass extinction events. So in long run, few more million years later, biodiversity could be okay, even we had 3 to 4 degree increase in the coming, say, 100 years. Would you agree this argument?
Yeah, it's an important point. And we have basically two different time scales. One is shallow time, ecological time scale, and the other is deeper time, evolutionary time scale. today I'm mostly talking about ecological time scale. So there is no much species extinction really, and no much species origination, speciation as well. So the system is explained by ecological change, like a temperature related distributional shift and tracking to thermal niche, etc. So in such time scale, something I talk today will be true. But in deeper time, like as Kenny said, 1 million years ago or much older, like tens of millions years ago, we have not only ecological range shift, but also extinction and speciation. In such case, we should be careful to separate ecological driver and evolutionary driver. We may see similar, but we may see different. And Kenny's point, like 1 million year future, we may have a diversity recovery. Yeah, it could be. Yeah, but I think it like a government or people are worrying about not 1 million year future, but more like 100 or 1,000 years, like a generation, time of our children's generation or grandchildren's generation. So in such case, yeah, tropical diversity decline could be quite true.
Great. Then I think there's one more question in the okay, it's more coming, but let me read this first. So since the temperature in deep sea is pretty low for deep sea, deep ocean, how could climate change affect the distribution pattern of deep sea biodiversity?
Yeah, it's also interesting point. I also do similar research in deep sea. And it's true, deep sea temperature is cold, but at the same time, that means they are quite adapted to cold, stable temperature environment. So they're kind of super sensitive to even small changes in temperature. And so, yeah, future climatic warming probably or very likely affect deep sea ecosystem and biodiversity as well. And the future warming changes will change not only temperature, but also like ocean circulation. So deep sea ecosystem could have substantial change.
Thanks for this question, Yuan Yuan. All right, there's another question asking, could you please kindly give some perspectives on the formation of new organisms and distinction of some important organisms that could likely play a key role on biogeochemical cycles in the past and how to project the future? Oh, big question. Yeah.
Yeah, it's a big question.
Yeah.
Yeah. For example, if we think like tens of millions or hundreds of millions of years time scale, we had a different silica producer. Now, the main silica producer is diatom in the present day ocean. But in super deep time ocean, silica producer was radiolarians instead of diatom. So in some timing, radiolarian went down and diatom came up. Such kind of evolutionary change should have substantial changes in silica cycling. It's one thing I can immediately think about.
All right. Thanks again to all the audience for your attending this talk at EEH, and thanks for the audience who asked questions. And last but not least, please thank our today's speaker, Moriaki. And yes, I think that's time is perfect. Now we can close. And as Moriaki said, if you are interested in any of those topics, please feel free to contact him. And he is now in Denmark. And we hopefully will meet again, Moriaki, in Denmark before you go back. Yes. All right. That's it. Bye-bye. Good evening and good afternoon.
Yeah, thank you very much,
Bye-bye.
Elvis and everybody. Thank you. Bye.